Muy buenas tardes. Nuevamente agradecemos su amable presencia. Antes de que demos inicio a esta conferencia, le pedimos un minuto de su atención para el siguiente aviso de seguridad. Ante todo, es importante que en todo momento guarde usted la calma. Identifique los puntos de evacuación que se encuentran en las puertas a su derecha. Evite obstruir con sus pertenencias, pasillos y toda área de circulación. En caso de alerta, usted deberá situarse en los muros laterales y traseros de la sala, áreas de menor riesgo. Una vez que esta alerta termine, se le solicitará evacuar la sala siguiendo las indicaciones del personal del colegio, quienes le pedirán abandonar el inmueble por las rutas que han sido marcadas en pasillos, escaleras, hasta el punto de reunión que se encuentra ubicado en el exterior de este edificio, en la esquina conformada por las calles de Donceles y República de Argentina. Usted será asistido en todo momento por el personal de seguridad del Colegio Nacional. Podrá regresar hasta que se dé autorización por el mismo personal del colegio. Nuevamente agradecemos su amable presencia y que disfrute esta actividad.
Muy buenas tardes, a nombre de doctor Sarukhán, a nombre propio, pero sobre todo a nombre del Colegio Nacional, les quiero dar la bienvenida a esta primera sesión del año 2019 de los viernes de la evolución. Los 2 años anteriores habíamos comenzado en febrero, pero este año decidimos aprovechar muy generosamente la presencia del doctor Andrei Lupas, del Instituto Max Planck para Biología del Desarrollo de Tübingen, una autoridad mundial en estructura y evolución de proteínas, que también con una generosidad extraordinaria ha apoyado mucho la creación de un centro, el primer centro de hecho asociado al Max Planck que tenemos en el país, que está en Ensenada. Él estuvo allí visitando al doctor Edgardo Sepúlveda y cuando quedó claro que iba a poder coincidir en México con la posibilidad de tenerlo aquí hoy con todos nosotros, pues rápidamente el Colegio Nacional lo quiso invitar. Empezamos con el pie derecho, si me permiten ponerlo en esos términos, porque todos sabemos de la ubicuidad de las proteínas en prácticamente todos los procesos biológicos, todos sabemos de las complicaciones que tenemos para entender la forma en que estructuras tan complejas han surgido a partir de motivos funcionales más pequeños, y si hay alguien que tenga una idea clara de cómo pudo haber sido ese proceso, es precisamente el doctor Lupas. Él comenzó como bioquímico, interesado originalmente en biología marina, y luego encontró la verdad, desde luego, en la biología evolutiva, se dirigió hacia ella y nunca obtuvo un título de licenciatura ni de maestría, directamente obtuvo el doctorado. Después de unos años en Princeton, regresó a Alemania y directamente como director en un instituto Max Planck, que como todos saben son centros de referencia en la actividad intelectual y en particular en la actividad científica. Para el doctor Sarukhán, para mí, para el Colegio Nacional es un placer tenerlo aquí, pero también es un placer enorme tenerlos a todos ustedes. Y es un placer enorme tenerlos a todos ustedes aquí porque, como ocurre, a mí siempre me conmueve mucho que la edad promedio del auditorio es baja, lo cual significa que la actividad del colegio está incidiendo sobre estudiantes y gente joven. Y además me alegra mucho que empecemos también este año porque, entre otras razones, como todos ustedes saben, hay unas confusiones grandes sobre el nivel de apoyo que se están dando a las actividades culturales y a las actividades científicas en particular. Y creo que el hecho de que haya tantos jóvenes el día de hoy aquí es una demostración de la confianza que todos compartimos en la importancia de las ciencias naturales y exactas para configurar una cultura nacional. Thank you so much, Andrei. The hall is yours.
Thank you. and I apologize for not being able to return the greeting in Spanish. But next time I come, I hope my Spanish will be functional. Right, so today I would like to tell you about a question that has preoccupied me for 20 years now, which is the question of how proteins came to be folded. And in the course of this lecture, I hope that I will convince you that it's a completely non-trivial problem and that it holds the key for one of the very important transitions in the origin of life. So allow me to start with a slide. I've been accreting and changing the slide for certainly a decade by now. Above the line, you will see in black geochemical and astrophysical events in the life of the Earth. And underneath, in blue, you will see major events of biology. And there are 2 points that I would like to point out to you. I mean, this is a complex slide, and one could spend an hour discussing it, but really there are just 2 points I want to make. One is that life progresses by catastrophes, and I mean true catastrophes, where 90% or more of all extant species are wiped out, are dead, right? And 2 of the biggest catastrophes that have been visited upon the Earth are both due to the release of oxygen, which is one of the strongest cell poisons we know. So the first major release of oxygen is known as the Great Oxygenation Event, and it happened about 2.2, 2.3 billion years ago. And during that time, the Earth had frozen completely. It was a massive ice age in which the entire surface of the Earth had frozen. These episodes are called Snowball Earth, because the entire Earth is covered by snow and ice. And this Snowball Earth coincides with the Great Oxygenation Event. And I think it's reasonable, although of course correlation is not causation, I think it's reasonable to assume that the freezing degassed the oceans and released oxygen that had been accumulating through the action of photosynthetic organisms. We had a certain Snowball Earth episode at this time here. Actually, there were 2 in very short succession. And this also coincides with the second major increase in oxygen levels, which brought the atmospheric oxygen to the levels we know today. And so, in essence, if we look at what happened on the biological side during these major poisoning events, in one, we probably have the origin of eukaryotes. Certainly in time, it coincides with this period. And it is quite reasonable to assume that the eukaryotes, which were probably a marginal form of life preceding this massive increase in oxygen concentrations, suddenly became very competitive and took over after more than 90% of all other species being wiped out. And we see another huge event here with the advent of modern oxygen levels. Multicellularity could increase tremendously because there was now enough oxidative potential in the gas that was being inhaled to feed a large body. And so instead of being unicellular and having to constantly exchange gases across the cellular interface, suddenly it became possible to have multicellular anatomies and to feed all the cells with oxygen through a central system, respiratory system. And so we see very shortly after this the origin of multicellular animals, plants, and fungi. And not too long after that, the colonization of the dry ground. Until then, everything was in the sea. All right, now this is one point that I wanted to make. Life progresses by catastrophes. The other point I would like to make is that the last common ancestor of all living beings on Earth is dated roughly to about 3.5 billion years ago. And so from the first point where we had liquid water on Earth to the last common ancestor, we had less than 1 billion years. So in this 1 billion years, we evolved from basic organic chemistry to a full complement of biochemical reactions which are modern. Essentially, the last common ancestor could do most of what we can do today. Most of the enzymes that we use today were already in use back then. Most of the prototype of the folded proteins were in use. The transcription machinery, the translation machinery, everything that makes our cells run was already worked out, the genetic code. Everything was worked out at this time. So we are in desperate need to try to understand how it was possible to progress from seemingly nothing to life in such a short time. And today I would like to focus on one specific event in this time, which is the transition from a precursor world, almost certainly an RNA-based world, to the DNA-based world we have today, right? Our cells have their information encoded in DNA. RNA mediates from the DNA to the protein world, and then the proteins, which are encoded in the DNA, do the entire catalytic reaction system in order to promote life and to allow it to reproduce. All right, so what kind of molecular innovations would we have needed in order to get to the systems that we have today? And it's fairly clear we would have needed metabolic networks in order to start making the building blocks of life. We would have had some of these building blocks associate into polymers, long chains, such as DNA, such as polypeptides. Then we would have had these polymers associate into networks capable of producing a network behavior that allowed them to transmit information and reproduce themselves, right? And therefore, at this point, we would have gotten the first replicative systems. Then we would have needed compartmentalization, what today we call cells. At some point, we would have had to compartmentalize the reactions that were happening. We would have needed a code in order to allow the information-carrying molecule to specify all the other parts of the system. And in the end, we would have needed equal cell division because we need a reproduction in which information is duplicated without loss. And here I will not go into greater detail with these. I would just like to say that at this point, where polymer networks became capable of autocatalytic replication, this, in my mind, is the beginning of life. This is where organic chemistry made the transition to biochemistry. And the reason I say that this is the beginning of life is because this is the simplest step at which I expect that we can have Darwinian evolution. And Darwinian evolution relies really on 4 criteria, which are summarized in these 2 statements, which I'm sure you are familiar with: descent with modification and survival of the fittest. Descent means that you need to have offspring. Modification means that your offspring will not be identical to you. There will always be changes happening as time goes by. Survival means that not all your offspring will live to reproduce. And of the fittest, well, biologists have an awfully hard time defining what fitness is. I can't tell you what fitness is. All I need is that the survival is not random, right? In other words, that there is a tendency to weed out certain aspects and enhance other aspects. And once you have these 4 elements together, you have a system which is capable of evolving into a direction in which there's an evolutionary pressure. And it will get, over time, optimized for the kind of environment it has. And then, as long as the environment doesn't change too dramatically, the system will do quite well. And that's why I say biology progresses by catastrophe, because the environments have to change radically in order to allow new ways of doing things to come up. Okay. Now, In the history of molecular biology, people started noticing very early on that whenever they were looking at a basic process in the cell, the information was encoded in DNA and the catalytic activity was in proteins. But everything in between was mediated by a molecule called RNA, which is chemically similar to DNA, but is now essentially just a mediator. DNA in normal cells, DNA does not encode information, it transmits information, and DNA is itself barely catalytic. It has one essential catalytic function, and that is making protein. That's what it does, right? And this centrality of RNA and the ability to both carry information and catalyze certain reactions led people to claim that these central RNA molecules are the survivors of a precursor world to our DNA world, and this world they called the RNA world, right? And this term you probably have heard. All right, and I would like to argue that the RNA world in that form did not exist. From the beginning, the RNA world was an RNA peptide world. That is, there are a number of things RNA cannot do, and peptides, which were probably abiotically already in all the mixtures at the time, were able to provide certain functionality that the RNA could not have. And I have tried to list some of the functionality here, and I don't want to read all of it, although it's probably too small for many of you to read it. But in fact, what happens is that peptides are able, even without any structure or any great length, to chelate molecules, for example, metals. They are excellent binders of nucleotides and nucleotide-derived cofactors. They promote the thermostability of RNA by intercalating into the grooves of the RNA. They promote the folding of RNA by disfavoring certain unproductive intermediates. They support the adhesion of RNA to surfaces. RNA doesn't adhere well to anything. Peptides stick like crazy. Some of the strongest glues that we use today, chemically derived glues, are derived from peptidic material. And historically, it used to be that we boiled bones in order to get glue. Right. And many peptides are membrane active. They are able to interact with membranes, to insert into membranes, to functionalize membranes, to make pores through membranes, to divide membranes. They are able to do a lot of things that a very primitive RNA-based organism would have needed to do, and which RNA cannot do. And so, to me, it seems fairly clear that the peptides were there from the beginning. And so, I would contend, without wishing to spend more time on this point now, that the origin of life is approximated quite clearly by this drawing by Maurits Escher. One hand is the RNA, and one hand is the peptides, and one is making the other, so that the cycle of amplification between peptides and the RNA led to the first self-replicating and fully functional networks. And so at the bottom you see here the last sentence from The Origin of Species by Charles Darwin. And in my opinion, from so simple a beginning, endless forms most wonderful and most beautiful are being evolved. And that includes all of us. We're all the result of this interplay of nucleic acids and peptides. And so now let me come to the point at which I started my work. And that is the question of the DNA protein world, which we live today. Our world is the DNA protein world, and our reliance on folded proteins. And the question, how did this transition from a primordial peptide world to the modern protein world occur, right? And one of the questions is, of course, why did it happen? And I've already told you that, in my opinion, life progresses by catastrophe. So something must have happened that wiped out the RNA world almost completely. And one thing that the RNA is much more sensitive to than polypeptides and DNA is temperature. And so, in my opinion, there has been the proposal of a major impact at the end of the late meteorite bombardment, which is variously called the late veneer. And this must have been a truly staggering impact, which, in my opinion, could have raised the temperature on Earth sufficiently to disfavor long RNA molecules and allow networks that were using the much more stable DNA for information repository to come up and take over life. Now, that is all very fine, but a problem is that the RNA was also doing catalysis. And you do need the catalysts, and the DNA can't do catalysis. And so the question becomes, if the RNA went away, how did the peptides, which were cofactors of the RNA world, assume the ability to do catalysis? And in essence, they could only have done that if they had increased the complexity of their chains sufficiently to achieve folding. And this is really the topic that I would like to field today. And so, why is folding so important? Folding is important because proteins cannot develop catalytic activity if they do not have a defined three-dimensional structures. So you can do some things with proteins that are not fully structured or even completely random coil, but if you want to do chemistry, you have to have structure, right? And this structure, it turns out, is a very complicated property. So nucleic acids fold quite spontaneously. If you do a PCR reaction, you melt the DNA, you reanneal it, you melt it, you reanneal it, and you'll still have your DNA. At the end of 100 cycles, you'll still have the DNA there. You can do these cycles with almost no loss of material. With proteins, you cannot do it, right? You take an egg, you put it in the pan, you get omelet, you cool it down, you have cold omelet. You do not get your egg back. Proteins, once they have coagulated and formed aggregates, have to be degraded and resynthesized. Okay? All right. And so you could say, well, okay, proteins are easily denatured, proteins lose their structure easily, and so on and so forth. But they are all folded anyway. Any polypeptide chain has a structure. And it turns out that that's not true. There have been a fairly large number of selections and scans being made for folded exemplars within random peptide libraries, and the success rate has been tremendously low. Typically, if there was any success at all in an experiment, there was maybe 1 or 2 folded exemplars per library of 10 to the 9th to 10 to the 12th peptides, polypeptides. And so I would say that only an insignificant proportion of any random chain of amino acids will have a folded structure. And so nature could not have progressed from peptides to folded proteins just by screening through random chains. It would not have worked. The likelihood would have been much, much too small, right? And so how did nature get around the protein folding problem? How did nature find all these folding proteins and find them quite efficiently? Because as you saw, the last common ancestor is a long, long time in our past. So by that time, most of the families of folded proteins had been discovered and established. All right, and so when I started my work in Tübingen, I proceeded from the assumption that the first proteins evolved from these peptides of the RNA world by an increase in complexity of the peptides. Now, these peptides, in order to be active with RNA, would have had to bind specifically. And specific interaction requires the exclusion of water. And so when you exclude water from a peptide, it starts forming what is called secondary structure. So either such helical arrangements or these, what we call beta strands. So if you exclude water, this is what you will get. And so the RNA must have promoted the formation of secondary structure. And in fact, the experiment can be done today. You can take a large RNA molecule like a ribosomal RNA and add a random peptide library to it. And you can spectroscopically find that this RNA will induce secondary structure in your random library of peptides. So because the natural polypeptides have an affinity, an intrinsic affinity for RNA, and because the binding to the RNA excludes water, you are starting to produce secondary structure. And so as these secondary structures grew longer, they became less and less dependent on the RNA scaffold in order to produce these kinds of local structures. And once the RNA was gone, if they were long enough, they could start folding up on each other, and they would not need a scaffold anymore. And that was the step that went from long peptides of the RNA world to autonomously folded proteins of the DNA world. All right, and so where do I come in? I said that if this idea is true, the traces of these peptides should be present in the proteins of today. Just like linguists can reconstruct ancient languages by studying modern languages and comparing what they have in common, we could study what the ancient peptides were by comparing modern proteins and trying to ferret out where the roots of specific families are. And in doing this, I used the sequence of proteins and not the structure, because biophysics makes the structure of proteins, especially at the local level, converge very quickly. It's the sequence of proteins which has a space that's almost infinite, and so convergence almost does not occur, right? For this, just consider that the average protein domain, so the average unit of protein, which is currently in our structure database, has about 185 amino acids. And so the sequence complexity of a chain of 185 amino acids is 20 to the power of 185. Now, I don't know if you know the number of particles estimated to exist in the entire universe. It's about 10 to the 82nd. And the number of seconds elapsed since the Big Bang is about, I believe, 10 to the 17th. So 20 to the power of 185 for all practical purposes cannot be distinguished from infinity for us. And so the sequence space of proteins is practically, is functionally infinite. And therefore, we should not see convergence. We should be able to study the sequence of proteins and use very, very powerful statistical methods to try to find any commonality that they still have. All right, and that's what we did. And the parallel to language, I didn't make it by chance. It was actually at the basis of my thought. When I started thinking about this, I was working at SmithKline Beecham, a pharmaceutical company, and my boss was a computational linguist, David Searles. And he was very interested in how biology and human language were going in parallel. And he, in fact, became quite well known for showing that the Chomsky hierarchy of grammar could actually be applied to DNA. And so he asked me, well, how does the Chomsky hierarchy apply to protein? And I said, I don't think it applies at all. And in fact, discussing with him, I came to realize that the parallel between proteins and language is in the vocabulary. The words of our language evolve the way proteins evolve in nature. And so I would like to give you here an example of how I view the process as functioning, and then I will show you an example from the protein world, which does exactly the same thing. So allow me to start with this root here. It's a very ancient root. We know it from cuneiform tablets from Sumer. The syllable was probably pronounced kane or something like that. It meant reed, you know, a plant in the swamp. And kane, through a variety of Semitic languages, was acquired in Greek as kana, in Latin also as kana, a very surprising thing which no linguist can explain to me, but it was. And via the French can, it came into English as cane. And so the English word cane still means reed, right? Sugar cane, it's still a reed. And so we have here 5,000 years of language evolution in which neither the sound of the word has changed much nor its meaning. Right. Now, in Greek times, we have several paralogies. Paralogies meaning that the word was duplicated and acquired a new meaning, a new function separate from the original function. So the Greek used the reeds, which were very long, straight, and rigid, and had very nice nodes at specific intervals, as measuring rods. They would measure distances with it. And they called these rulers kanon. And from that, they also called the abstract concept of a rule, canon. And from this comes the church canon, the church rule, and the canon as a musical form where you sing by rules. Here, the Greek used the flexible properties of very young reeds to weave baskets, and they called them canastron, from which comes the canistrum and the canister and canasta and a variety of words from this area. And the Romans realized that the reeds were hollow, and if you punctured the membranes, because they were tapered, you could push them into each other, and you could put water through. You had a water pipe ready-made just by cutting reeds. And they called these canalis, from which comes the channel and the canal and the canyon, and also by derivatives, the canon and the cannelloni and the cannelure and a variety of other words. And so if we look at the history of this word, we see that a TV channel and a church rule have the last common ancestor in a Sumerian plant. And this is what happens in the protein world in pretty much the same way. So let us look at proteins. So here we have a piece of a protein, a peptide, which we have reconstructed by comparing modern proteins, and which we propose existed at the time of the RNA world. And this peptide gave a number of offspring which bind RNA. This one is part of the ribosome. This part is now autonomously folded outside the ribosome, not in contact with the RNA, but it's part of a particle that degrades RNA. It's the so-called exosome. All right. And so we think this is the orthologous activity, the ancient activity which is binding RNA. But here, through a duplication of this piece, we got a pocket forming, and this pocket is really good at binding biotin. And so all the biotin-dependent enzymes in biology today are derivatives of this ancient cage that evolved by duplication of this peptide. And then we have two other things where we see this fragment assuming structural roles, which we are in the process of defining more and more specifically, but I will not go into the biochemistry of these. But in any case, I hope that you can see the parallel between this and this. We see a lot of paralogy happening, a lot of decorations, and a lot of changes in the meaning, in the functionality of these entities. All right, so how did we do our work? we essentially compared all the proteins of known structure in their sequence, and we tried to find pieces that were similar locally, but not globally. So the pieces only had a small part of overlap where they were the same in structure and similar in sequence, right? And then after we had these local matches, we used their structure to validate that they were indeed the same thing. And for this, we built a Bioinformatics Toolkit, which is public, and I would be very happy to learn that you are using it. We made it completely open so that other people can work with our tools. The core of this toolkit is a program called HHPRED, which allows us to look back in time to make statistical inferences back in time more than 3.5 billion years ago. So this is the core of the method that allowed us to define the fragments that I'm going to show you. But before I get to the complete vocabulary of fragments, let me show you what our data look like. So here, for example, you see these 8 proteins, and they look different, and they are different, and they do different things. But they have one thing in common, and that's this little red piece. This little red piece binds nucleic acids. That's what it does. And it brings the nucleic acid to the rest of the protein, which then does whatever it does with the nucleic acid. And so this little piece here, the red piece, I have superimposed all of them here, and you see that they are still very similar. After 3.5 billion years of evolution, they're still pretty much the same thing, just like Kane, right? And here are the sequences of these fragments, and these are the ones where we could see, just by analyzing the succession of letters, that there was a statistically robust signal of relatedness in there. And so we claim that this fragment, which is called the helix-hairpin helix motif, was one of the ancient peptides which existed at the time of the RNA world and was working with the RNA in a functionality that right now I can't tell you what it was, but we're working to try to figure out. And these are the 40 fragments which we have reconstructed. So based on this way of analyzing sequences and looking for local similarities in globally dissimilar environments, we have found these 40. We have more, of course, but the statistical support is not as strong as for these. So at some point we put a line and said, these are our best ones, right? We had to get past the reviewers. So we had to show them our best cases. And these are our best cases. And so I will not spend too much time on these, although really I could spend my whole lecture just on these. They're really an amazing array of things. A few remarks about them. The first is that most people, if you tell them of peptides, they will think of something like this, right? Which is a piece of protein which you see all the time. But stuff like this, you don't see anywhere. How can this be ancient? And it is. It is. It occurs in several protein folds at the core of the fold. But it's not something that in and of itself might ever be close to folding. It does require the RNA, right? The other thing that we find is all these dotted ones, and there's a lot of them, they form their fold by amplification. If you take this, for example, if you have 2 copies of it, it will fold. We've done the experiment and we know it's true. If you take this, you make 2 copies of it, it will fold. If you make only 1 copy, it will not fold, right? This one, 3 copies, and it will fold. This one, 2 copies, and it will fold. And all the ones that form their folds by this amplification are dotted, and you see there's a lot of them. And I will show you some of the experiments that we've done with this. Another thing, all the ones that are with a yellow background interact with nucleic acids. And what I mean is we have high-resolution structures which show that this fragment interacts with nucleic acid, right? Okay, so it's a very high bar that we have set. All of these bind nucleotides. All of these coordinate metals, including iron sulfur. So the kinds of functions that were essential in the RNA world are very highly represented in our fragments. Now, if we take the 40 most frequent fragments in nature today, the most frequent ones, not the most ancient ones, there's one nucleotide binder among them, there's 2 nucleic acid interactors, and there's 4 metal binders. That's all. So we're tremendously enriching for functionalities that would have been relevant in a peptide world. Now, let me show you some experiments that we have done to try to explore how these pieces could have given us folded protein. And so let me show you what we've done with this thing, which is called a blade. The blade is called that way because the biggest fold that it makes is the so-called propeller. It looks like a propeller, right? And the propeller contains multiple copies of the blade arranged radially. Okay. And this blade can also make other structures, but we were interested in this relationship. And we were wondering, is it enough to take one blade and it will associate to make a propeller? Or do you need to do more things with it? And the answer is that we looked for a while to find something in nature that would allow us to do the experiment in a fairly straightforward way. And in the end, we found this protein here. I don't know how many geneticists are among us, but you can see that this protein underwent almost no mutations. All the mutations are yellow, and there's practically none of them. Everything else is the same, one copy after the other. There's 14 copies of the blade, 14 blades, and there's almost no mutations. When you look at the structure, this is the crystal structure we determined, you can see that the mutations are almost all in the binding site of the propeller. So this propeller evolved very, very recently and is moving towards a functionality. And if you look at the DNA, you can see that there are no synonymous mutations. There are only non-synonymous mutations which gave rise to these point mutations. So if you were to study the evolutionary pressure on this by some dn over ds measure, ds over dn measure, you would essentially get infinity. All right. So what did we do with it? We took one blade, this one, and we asked, can this one blade associate to make this propeller? And the answer was not. We could not do it. There was no way we could go from here to here. But as soon as we took 2 blades, here, 2 blades, we got a structure. This thing folded very efficiently, and it gave us an 8-bladed propeller, right? There's 4 chains of this in here, and they make an 8-bladed propeller. So then we took 3 blades. Now, 3 blades cannot make an 8-bladed propeller, and they don't. They make a 9-bladed propeller. Okay? Then we put 4 blades in a row, right? Now, 4 blades can make an 8-bladed propeller, and they do. But you see the symmetry is different from the previous one. Okay? So this is the second 8-bladed propeller we got. And then we put 5 blades in a row. And we could see biochemically there was a dimer. There were 2 chains in the structure. And so we thought we would get a 10-bladed propeller. But we didn't. We got this. Now, this is a homodimer. The 2 chains in this molecule are identical. But the chains don't look the same. This one makes all 5 expected blades. This one makes 4 blades, and one blade turned into an alpha helix. Also, it's not a propeller. A propeller, if a propeller is like this one's like this. The two halves interact differently from the way they would in a propeller. This is not a propeller. And we can see why the helix is here. It's there to close a hole that has opened in the structure. So you could say it's a monster. And it is. Maybe some of you know about Richard Goldschmidt and his theory of hopeful monsters, where developmental processes with small changes might give you a macroscopic change, a strong phenotypic change, like 2 more fingers. And he called these hopeful monsters because they were monstrous, they were different from everybody else of that species, and they were hopeful because they were hoping to inherit the Earth. And if there's a catastrophe and everybody who has only 5 fingers dies, then the few people who have 7 fingers are going to take over the Earth. And soon enough, all humans will have 7 fingers, right? And so that is what in Richard Goldschmidt's idea was a hopeful monster. And this, I would say, is a hopeful monster. It's a piece of a propeller which is very stable, it's very friendly, and it's looking for a function. And we're trying to give it one, but so far we haven't succeeded. All right, so let me show you another one that we've played with, and that's this one. And this one has a bit of a longer history. So in essence, this is a ribosome, right? The large subunit, the small subunit of the ribosome, and in particular what we have highlighted here are the proteins that are in the ribosome. And if we look at the center of the ribosome, we can see that the proteinaceous material pretty much doesn't have secondary structure. It's just chains. There's no structure. Now, if you go further out, you start seeing structures forming. You still have a lot of unstructured material, but you do have structured material coming up. As you go out further, you see proteins that are now pretty much fully structured. Although all the ones that have been tried, as far as I know, in this circle, do not fold without the RNA. They look like they are proteins, but they are not. And then this is the whole complement of proteins. And you see out here, we have a number of red fragments. These are the fragments we found as having given rise to the folded proteins. So one could claim that in essence, the folded proteins of the cytosol are the offspring of the fragments that were in the outer shell of the ribosome. They were the ones that were able to make the next step. They were complex enough and close enough to a folded structure to be able to take the step into the cytosol and become true proteins. And so we've been interested in this one. Right? We think that this is a progressive emancipation, and whoops, where is it? There it is. Okay, and we've been interested in this one. Can we take this red piece here and make this protein here? Right? And so this is the piece. It makes all sorts of different proteins, and one of these it makes by repetition. And that's the one we're going to try to make. We're going to take this piece, we're going to repeat it 3 times, and then we're going to hopefully have something that folds. Right? So these are the actual proteins that we took to make our constructs. And this is the design procedure by which we arrived from this fragment to this fragment by mutating only 2 residues, these 2. If we change these 2 residues, then we get this structure. The crystal structure we solved is in color, and the structure we thought we would obtain is in gray. And you see that the color and the gray agree quite well. So in essence, we could say, yes, we have made the soluble protein by using a piece of a ribosome with minimal changes. And this is the full protein we made. It looks different in one specific respect, but I will not go there because it would take me way too much time. And so I would like to come to, no. I would like to come to this piece right here. So I've shown you two experiments that we've done to see whether we can get folded material from unstructured polypeptides. And in principle, we can, and we are doing this with a number of others. We're looking for other processes. Amplification, I think, is really important, but there are other processes which we will have to explore. And we are, I think, going to nail this within the next decade and a half. We'll have a pretty clear idea of how we go from unstructured to structure. Now, one of the problems that I'm starting to work on now, and which I think is of overriding importance, is not to explore more of the step from unstructured to structure. I think that we have that under control. I would like to understand now what use were the unstructured peptides to the RNA before the transition to the DNA world. What were these peptides doing in the RNA world? And we can do this experimentally, and we've started to do it. We're trying to find systems in which the peptides that we have identified are useful and help to produce a favorable outcome in the RNA molecules involved. All right, and so I would have one more point, and then I will stop. And the point is, what about evolutionary scenarios? So far I've only told you this is related to this. But most of the time when you publish a paper, people want to know, well, how are they related? That's a very different story, right? Saying that this is related to this doesn't necessarily let you know at all how this became this, or whether this became this and not this became this, right? And so scenarios are a huge problem. But unfortunately, we have to make them, right? So I didn't make this voluntarily, but the reviewers forced me to draw out what the relationship of all the barrels were that we had characterized. And so I drew this out, right? And do I believe this is the truth? The answer is no. I think that scenarios are much closer to fiction than these statistical inferences of homology. And I would like to leave you with how I think about this, right? You are aware of these little games where you connect the dots. If you follow the numbers, right? It's very good for children to learn how to count. And if you connect them correctly, you get a beautiful picture. Now, in our studies, we don't get the numbers. That's the first thing. The second thing is, most of the dots are extinct. And so the picture that we draw might not reflect very accurately the origin of the material. Right. And with this, I would like to thank the collaborators with whom I have worked on this project for 20 years now, and I'm very happy to answer questions if you have them. Should I put this away or?
Andrei, thank you very much for this enticing, fascinating, provocative lecture. it's quite impressive how you can go backwards, and at the same time, it's wonderful to see those huge set of open questions,
Yes.
which is perhaps the best stimuli for the students and the young scientists here. Tenemos tiempo para preguntas, comentarios. Don Omar, sí.
Buenas noches, doctor. Una de las preguntas que me ha llamado mucho la atención es la relación que existe entre la biología y la acelerada progreso de las computadoras. Entonces, la pregunta es concreta, si en este momento ya con la capacidad tan asombrosa de la memoria de las computadoras, se pueden predecir plegamientos de una proteína de 600 residuos, si ya es capaz de calcular algorítmicamente las computadoras todas las posibles combinaciones que puede dar una proteína de 600 plegamientos, de 600 residuos en su conformación tridimensional. Si me permite otra pregunta, sé que hay proteínas.
I think it would be better to do one question at a time.
Okay.
Yeah. Right. Right. So.
¿Me permite una segunda pregunta o ya nada más con una?
No, nada más, vamos pregunta a pregunta, pasito a pasito.
Gracias. Entonces, nada más esa de momento.
Por lo pronto.
Okay, so if I understood the question correctly, it was, can we predict the structure of a protein formed of 600 amino acids? And the answer I would give you is we cannot predict the structure of a protein made of 200 amino acids or even 150. So there is a competition every 2 years. It's called CASP, Critical Assessment of Structure Prediction. And in it, experimental scientists give the sequence of a protein that they have solved, but not the structure. And bioinformaticians can predict the structure of the sequence. And they typically have 2 weeks for a target, then the target closes. And at the end of the year, everybody gets to see the predictions and the true answers. And we're making progress, but the progress is very small. So this has been going on for, I forget, 14 rounds. And we do see progress, but we still cannot predict in a way in which you would say, like an engineer can predict the stability of a bridge, right? We cannot tolerate error there because then the bridge would collapse. In the protein world, we have to accept the error because the best predictions we can make are not really always very accurate. Now, with respect to the computing power, our computing power is not impressive. I'm sorry to say that, but I already said that the sequence complexity of a chain, of a median chain in the protein data bank has a sequence complexity that's vastly larger than the number of particles in the universe. Now, if you start combinatorially, you very quickly exhaust all the computers in the world without getting anywhere. It's not going to work, and our computing power is not nearly massive enough to even solve simple questions.
Sí, ¿había una segunda pregunta de la misma persona o...?
Muchas gracias por la segunda oportunidad. Hay proteínas que sirven como, digamos, chaperonas o chaperoninas para ayudar a plegar a las proteínas recién formadas. ¿Esto no nos llevaría a una especie de qué fue primero, si las cofactores o el plegamiento de las proteínas? O sea, hablo de una familia de proteínas que cuando la secuencia lineal de los aminoácidos recién sintetizados sirven como cofactores para que ayuden a estas proteínas tanto a plegarlas y tanto a llevarlas a su funcionamiento normal, que inclusive les ayudan a atravesar membranas. ¿Qué relación hay entonces entre estas proteínas que ya tienen esa función y la función del plegamiento de las nuevas? Gracias.
Yeah. Okay, if I understand correctly, it is the question on whether protein-based chaperones could have helped the first steps in helping other polypeptides getting to the folded state and to manifesting their activity. Is that right?
Yeah.
Yes. Okay. So, of course, from my talk, you saw that I'm implying that the RNA was the original chaperone. Polypeptides were folding on the RNA and manifesting its function. And so, when making the step to independence, which ones were the first ones that were useful? And I fully agree with your thought that chaperones would have been extremely useful to have right up front. So polypeptides which were capable of folding autonomously, robustly, and to help others to fold after they themselves had folded. And that's why I showed you the two that I showed you, because beta propellers and TPR repeats are known today to have a primary function in binding unstructured polypeptides and preventing their aggregation, which is the most important step in allowing them to fold. And so, yes, we are thinking along the same way as you are. Unfortunately, none of the molecules we made in our experiments has chaperone function. So they fold autonomously, but they can't help other molecules to fold. So at some point, we'll probably get there.
Había otra pregunta por don David. Sí, por favor.
Hola, buenas tardes. Bueno, es como, ¿cómo decirlo? Bueno, los plásmidos sabemos que son RNA como fraccionado en bacterias. Y comentaba que en el RNA encontramos estas zonas que permiten el plegamiento de proteínas. Entonces, ¿también se han encontrado estos péptidos que ayudan al plegamiento de proteínas en los plásmidos? ¿O solo lo basaron con el RNA completo? Gracias.
Okay, so can okay, yeah, just for that. Yeah, okay. But the question, as far as I understand it, is valid because there are RNA structures which would favor specific. Yeah. Yes.
Nada más una aclaración. Hasta donde sabemos no hay plásmidos de RNA en bacterias, son plásmidos de DNA, pero ese DNA puede estar codificando para RNAs funcionales, ciertamente, ¿no?
Mm-hmm.
So, please.
Yeah, so, stepping one step back from the question you posed is, are there any nucleic acid motifs that could engender folding in peptides, could produce useful complexes, or do you need the entire RNA molecule, like the entire ribosome RNA, in order to get individual polypeptides to fold on it? Is that correct? Is that what you were asking?
Yes, she was asking that.
Yes.
Precisely.
Yes. So, I find that a very difficult question, very interesting but very difficult. We have tried to relate our fragments with specific motifs in the RNA. But so far, statistically, we have found no correlations between specific RNA structures and specific protein sequences. So,
Don Omar, por favor.
Okay, what is the role of the ions and cations in the folding process?
In the folding process, right. There are polypeptide chains which require specific ions for folding. For example, the zinc fingers, right? Zinc fingers will typically not fold unless they have zinc. There are a few other ones which require magnesium or calcium. Most protein folding functions independently of specific ions, but it may require a certain ionic strength. So if you give the protein distilled water, it will probably denature. You will need to keep the protein in a salt medium in order to achieve the correct hydration shell that will allow it to fold.
If I may say something before we proceed to the next question, the example that you use of the zinc fingers will not be expected to be a very ancient one, because zinc is not so easily soluble unless we have lots of oxygen in the atmosphere, so they will come quite late.
Yeah, none of our ancient ones bring zinc.
Don Omar, sí, por favor.
Gracias. No, Adrián. I just want to ask you, because I love trees. If we right now have the information as to build a tree based on proteins, which ones are more ancient than the others? Also, is how to reconstruct evolution of proteins based on a diagram that we call a tree. Do we have enough information as to do it?
The main problem with building protein trees is that proteins are multiply polyphyletic. So proteins have many origins. It's not like life where you go back to the last common ancestor, but you will have many trees. For some protein families, we can build very robust, very good trees. For many other ones, we don't have enough information, I think in many parts, because too many exemplars have died out. And so, if you have only 2 or 3 dots, it's very hard to connect them correctly. Basically, the drawing that I had shown, the picture that comes out, may not reflect at all the original tree of that protein family.
Can I raise a question about that?
Sure.
Do you actually believe that those proteins have died out during these 4 billion years of evolution, or simply that the sequences have diverged so much that we don't recognize them as homologous?
Well, gene loss is the dominant form of evolution. And so, of course, whenever you lose a gene, almost always you lose a protein.
Regardless of the families of genes that we will have, in your view? Because normally we have, if you have a gene that has an adaptive value, you have very successful expansion of the sequence.
Right, so genes, for example, that encode P-loop ATPases are going to remain dominant in every organism. And so all the dots, many, many dots will have died out, but there's enough dots to make a very beautiful tree, right? Whereas some biochemical activities are very rare and only few organisms have them. And they have traded them laterally so that their direct relatives don't have them. And so there, I would say, most of the extant proteins are the remnants of a very big bushy tree, right? So yes, they have diverged beyond recognition, but that's because they were at the ends of branches that kept going, right? So horses, for example, it took a very long time to make a good tree of horses, because with the modern horse you had essentially, right, the donkey, the zebra, there wasn't much.
Don Omar, el maestro Bonfil, y luego ahí tenemos 2 preguntas más atrás. Don Omar, por favor.
Buenas tardes. Muy rápidamente, ¿has realizado algún análisis termodinámico y cinético del proceso de plegamiento de estas proteínas para saber cómo se compara con proteínas que están en la naturaleza?
Okay. Well, we haven't tried to make enzymes. That's really difficult. So, since we had the question about computational power, I can tell you that no one has made a true enzyme yet. We've made things that have 2 orders of magnitude better catalysis. A natural enzyme has like 6, 7, 8 orders of magnitude. No one's come even close to making an enzyme. Now, with respect to our fragments, what we can do is to look at the rates of folding. We can look at the kinetics in which these chains collapse into a folded structure. And there the kinetics, we have looked in 2 cases, the kinetics are indistinguishable from the parent proteins. So folding in our constructs, even in this hopeful monster, proceeds just like for any natural protein.
Martín Bonfil, por favor.
Since I am a science writer, I don't think I understood all of your talk, but I would like to clarify for me some of the things. I understand that you say that the relationship between RNA and proteins maybe began as a chaperone function for DNA to enable proteins to fold. I think, is that what you said?
No, so what I had tried to say was that the initial networks that were capable of replicating transferred the information through the RNA, but required the activity of peptides to be able to do so. And these peptides were active in direct association with the RNA.
Oh, yeah.
And therefore you could claim that the RNA was the chaperone of the peptides. But really the functionality of the RNA was not to fold the peptides, that was incidental to obtaining the activity that was needed to replicate.
I said it the opposite. And the other part, you went a little too fast for me when you talked about the ribosome. You said that the proteins in the ribosome maybe were related to this set of ancient folds.
Yes.
And you said something like they were the ones that survived. Perhaps you could repeat that little bit.
Yes.
And then you tried to produce one of the folds beginning with one of the ribosomal proteins, right?
Yes.
Thanks.
Right. Yeah, so basically, not the entire RNA world was wiped out, else we wouldn't see any RNA there today. And in fact, the first ligase for peptides is the core of the ribosome. So the ribosome in many ways is the entity which allowed this autocatalytic amplification and reproduction. In some ways one could say the ribosome is the core of life. And the ribosome survived this huge wipeout. The ribosome is the last survivor of the RNA world, right? And it still forms the core of our DNA protein world. So, yes, the steps I had tried to imply in what I showed was that the ribosome got more and more complicated and grew larger and larger. And the outermost shell of polypeptides, which were the most recent ones adhering to the ribosome, were also the ones that have the fragments we have reconstructed from the soluble ones. So the soluble proteins seem to be related to the outer shell of the ribosome. they don't seem to be related to the inner shells of the ribosome. It's really the outer shell of the ribosome, which seems to have been the one that was capable of making the proteins.
I can see Martin smiling broadly because his thesis was precisely on the ribosome structures many years ago. Don Omar? Perdón, justo atrás de Don Omar.
Hello, Dr.
Perdón, justo atrás de Martín.
Hello, Dr. Agibin Jesus. I have one question. If a structure determines the function of protein, what about multifunctional proteins where a structure can develop many functions?
I'm not sure.
¿Podría repetir la pregunta?
Sí. What about the proteins
Es que
that...
con el sudor se borra en las manos.
What about proteins that are considered multifunctional proteins, whose motifs have, one motif can have a unique function?
All proteins, simply by virtue of their complexity, have more than one activity. Sometimes these activities are used by nature and sometimes they are not. But every protein you could name me, I could show you that it has 3, 4, 5 different possible activities. Some of them will manifest themselves, some will not, right? So if you make the transition from these simple peptidic things to autonomously folding things, you're going to have one active site and then an enormous part around it. And that part is not inert. It also has its crevices, it also has its side chains, and it can also do things. And sometimes these evolve into doing new things, and then they're being used. At other times, we can screen them in the laboratory, but nature doesn't use the activity. We can see that it's there, but nature doesn't use it.
Sí, por favor, la señorita.
Good evening and nice to meet you, Doctor. Well, it's a little bit related with his question. Since I'm not very familiar with this kind of experiments, I would like to know if you could elaborate a little bit about how do you design and perform the experiments related with finding a function for your proteins, for your design proteins or proteins you don't know the function.
Yeah, that is a very difficult question. You know, there's a variety of ways in which to approach this problem. One is to find binding. Right? If you want to do catalysis, the very first thing you need is to bind whatever you're going to act on. And so you can make mixtures, 100 compounds at a time, and you can add them to your protein, which is labeled, and by an MR you can see whether the protein changes its NOEs. If it does, then it probably binds something in this mixture. Now you can start deconvoluting the mixture to find what exactly it has bound. And in this way you can get to the point where we got with our hopeful monster that it binds riboflavin. And then the trouble starts. We worked for 3 years without getting anywhere trying to figure out how it binds riboflavin. It will not co-crystallize, so we can't see the riboflavin. In the NMR, the NOEs are not interpretable at high resolution, so we can't see where it goes. We can start making local changes to the riboflavin to see which ones continue binding, which ones stop binding. That went on for about 3/4 of a year. At the end, we didn't really see a pattern. You can try to find something that you could select in E. coli. For example, not have an enzyme that requires riboflavin be able to bind riboflavin and give it this as a domain that maybe will bind the riboflavin. That failed. You can try many things, but most of them will not work.
Don Omar, por favor.
Hi. First, excuse my bad English, bye. I think this is a must to practice, so keeping in mind the 2022 useful amino acids that are used in the folding, do you think that any form can be built or exist restricted forms or conformation? The same for the functions. No?
Could you rephrase your question? I'm
Okay.
not sure that.
Okay, I'm nervous. Keeping in mind the 2022 useful amino acids,
Yes.
do you think that any form can be built or exist restricted conformation or forms?
Wait, you mean any protein structure can be made with the 20 amino acids? No.
Yes.
Yes, that's the question? Yeah. Well, no, I don't think so. But that's a guess, not a knowledge.
Mm-hmm.
The problem is it's extraordinarily difficult to prove a negative, right? So if we try to make something and we can't, we may just be too stupid. So we have, I didn't show it, but in the course of reconstructing an evolutionary scenario, we came across a structure which we didn't think should exist. And we redetermined the structure of that protein, and the structure that had been published was wrong. And so then we spent 5 years trying to make the wrong structure, to find whether there's any sequence of amino acids which will give us this wrong structure. And we couldn't do it. But as I say, that doesn't prove that it's not possible. It just proves that we failed.
Don David, por favor.
Hi. You said that peptides were adhesive. And my question is, why? Does it have anything to do with the functional groups inside the chain or the charges it has or something like that?
Yes. The side chains are the main adhesive aspect, although there are a few parts of the main chain which can be a problem. But the side chains, usually the hydrophobic side chains, will allow you to stick to things. And if you have enough flexibility to orient things in the correct way in order to mold the surface, like in the glycine tryptophan peptides or the glycine phenylalanine peptides, then you get extraordinarily powerful glues. So the muscles, you know, these animals which adhere to ships, good luck prying them off, right? they use sledgehammers because the glue is so powerful. Yeah, it's a function of the side chains and of the flexibility of the chain so that it can mold the surface.
Well, I think we have no more questions, but before we call the lecture off, let me very briefly tell an anecdote. When I was visiting professor at the Institut Pasteur, I was shown a real treasure, namely the pieces of cardboard that were used by Jacques Monod to slide them and see how they could align the different amino acids. All of you have played those with those little booklets in which you can put a fish face on a giraffe or a duck foot on a person, and those were exactly the things that were being used 70 years ago. And nobody would have expected that the development of computer science on the one hand and of molecular biology on the other could allow us to do what we are doing nowadays for all the limitations that we still have with the computers and all the limitations that we have with bioinformatics. It's still surprising how far we have gone. And there are two lessons to keep in mind about this. First of all, that it's a field in which you can have interaction between mathematician, computer science, life sciences, which is absolutely extraordinary on the one hand. On the other hand, no funding agency at the time would have given $1 or €1 for the potential interaction between computer science and molecular biology. So it's a lesson to be kept in mind. One always has to leave spaces open for new fields of science to develop. Having said that, I will turn into Spanish. Quiero agradecerle muy profundamente al doctor Lupas su presencia el día de hoy aquí en el Colegio Nacional. Creo que hemos empezado las sesiones de este año de la mejor manera posible, con las espléndidas preguntas que han hecho, con la espléndida conferencia que nos ha dado. De manera que thank you so much, Andrei, for your lecture.
Thank you.
Y si me lo permiten, déjenme de una vez anunciarles el próximo viernes de la evolución, que va a ser el jueves 14 de febrero, no porque sea Día de la Amistad, sino porque son los días en que, o del amor, una cosa así, sino porque es el día en que el doctor Lauren Graham del MIT va a estar en México. Y vamos a tener un diálogo entre él y yo sobre el caso Lysenko. Va a ser una conversación sobre esa tragedia tan compleja que afectó la ciencia soviética de una forma impresionante, cuando por prejuicios ideológicos se favoreció un cierto punto de vista de ciencia aplicada y se lastimó de una manera pavorosa lo que era una de las mejores escuelas de genética en todo el mundo. Alguna vez el profesor Oparin me dijo que quien lo conocía mejor que él mismo era Lauren Graham. Y entonces, bueno, vamos a tener la charla aquí a las 6 de la tarde del viernes de la evolución, el jueves 14. Y al día siguiente, para no perder los viernes, vamos a tener una mesa redonda sobre cómo se enseña ciencia en la Facultad de Ciencias a las 12 del día, donde va a participar la doctora Patricia Graham. Lauren está en el MIT, ella está en Harvard, y la mesa redonda sobre cómo enseñamos ciencia va a ser con la doctora Patricia Graham, con la doctora Ana Barahona de la Facultad de Ciencias, y va a ser moderada por el doctor Ranulfo Romo, miembro del Colegio Nacional. Entonces, la propaganda se va a hacer llegar como siempre, pero están invitados muy cordialmente a estas dos actividades adicionales. Muchas gracias, buenas noches.
Su amable presencia y también una calurosa felicitación a todos los biólogos en su día. Felicidades a los biólogos.